Welcome to Ask the Expert. This is a brief and informative and lively discussion, we hope, with experts in type 1 diabetes and related interdisciplinary research. We're recording this event. We're going to post it on the Sugar Science site, our YouTube channel, shortly after the presentation. And if you have any questions for our guest today is Dr. Roberto Mallone, please feel free to enter them in the chat or raise your hand at the end of the presentation. So today we have our guest, Dr. Roberto Mallone, coming to us from France, in Paris. Paris, Inserm in Paris. He's a team leader along with Sylvana Yu at the Institut Cochin at Inserm, and his team is really focused on T-cell tolerance, biomarkers, and therapies in type 1 diabetes. And he also has both academic and clinical appointments. So as we were discussing earlier, it makes for a very packed schedule, I'm sure.
Yeah.
He has a deep understanding of the immune cell landscape in type 1 diabetes, and he has just published a really deep dive, a very comprehensive and thorough review entitled T-cell epitopes and neo-epitopes in type 1 diabetes, a comprehensive update and reappraisal in diabetes in July 2020. And this review questioned the current, 10-year-old T-cell epitope catalogs, and using IEDB, which is the immune epitope database identifier, along with other tools, it really calls for the improved epitope validation. So I was really impressed with this careful characterization and grading of both mouse and human epitopes, and looked forward to I'm looking forward to having him walk us through the paper. And he also has another sort of thought-provoking paper along with Decio Isric that came out in Diabetologia, October 2020, which is presumption of innocence for beta cells. Why are they vulnerable autoimmune targets in type 1 diabetes? So I'm hoping that you can talk to us about both these really interesting reviews and your work. And so I'm just going to let you take it away from here.
Thank you, Monica, for the invitation and the kind introduction. I think it's a very interesting exercise today, and I will try to walk through these recent papers and the view that they open a bit revised about the pathogenesis of type 1 diabetes. So let me see if I can share my screen. I think we are good. Yeah. Hide floating meeting controls. This we put in the corner. Yeah.
That looks
so
great.
Today the title that I was given was T-cell epitopes and neo-epitopes in type 1 diabetes, and I think the key message of this work I'm going to present is that it's important to characterize epitopes, and I will not be speaking much about neo-epitopes, but we can discuss later on, not only to increase our catalog and the possibility of monitoring the immunity of the disease, but also to gain some insights in the pathogenesis of type 1 diabetes. So our key interest is in CD8 T cells in type 1 diabetes. Why is that? Because when you look at pancreas from type 1 diabetic patients, there are two histopathological hallmarks. The first is the upregulation of HLA Class 1 molecules. The second is the immune infiltration that is quite variable, but is dominated by CD8 T cells. And so this basically witnesses the crosstalk that is engaged between islet reactive CD8 T cells and beta cells that present in the context of these upregulated HLA Class 1 molecules, antigens, and these are in the forms of peptides derived from the beta cell inside for recognition by CD8 T cell. And this is what engages the T cell to actually kill the beta cell eventually through cytotoxic mechanisms. And our observation stemmed from this paper that we published back in 2018, where basically we took blood samples from new onset type 1 diabetes patients and healthy controls and isolated the peripheral blood mononuclear cells and went to look at the CD8 T cells that were recognized as islet antigens using highly sensitive tetramerases. The reason is the surprising observation that we made is that you see here we focus on CD8 T cells against the antigens that have been known in type 1 diabetes for a long time. You see ZNT8, preproinsulin, GAD, IA2, IGRP. And you see that when you look at the frequency between type 1 diabetes in red and healthy donors, this frequency is actually not any different with one single exception, irrespective of the disease status. And moreover, this frequency falls in a relatively narrow range. You see here that is highlighted with this gray band. And this narrow range actually, it's due to the fact that when you look at the phenotype of these CD8 T cells, actually you see that a large proportion of these T cells are actually naive, meaning that they are potentially autoreactive, but they have not really been engaged in the autoimmune process. So this was a quite striking observation. And the first thing we thought when looking at this data is that perhaps we were not studying the right islet antigens. Indeed, for decades, all investigators, including ourselves, have been focused on these antigens that have been characterized before, either as targets of human autoantibodies or in the NOD mouse. But we actually had no information about which of these antigens in the form of peptides are naturally processed and presented by human beta cells. So we devised a strategy in which beta cells that were exposed or not to inflammatory cytokines, mimicking the insulitis video of type 1 diabetes, were cultured. And then we purified these HLA molecules carrying the peptides. You see that these peptides come from the inside, from the digestion of protein, especially misfolded one through the proteasome, and are then loaded on the surface of HLA class 1 molecules. And so then we eluted these peptides to identify them by mass spectrometry. And this is how we obtained this first cartography of the antigen exposed by beta cells. You see here a snapshot of the source proteins of all the peptides we have identified in this work. And you see that there are a lot of the antigens already known, underlined here: chromogranin, insulin, this is IA2, GAD, ZNT8, although with a relative underrepresentation of, for example, GAD and ZNT8. But also we observed that there were a number of other antigens, and especially that antigens that are derived from granule proteins were particularly represented. About 1/3 of the peptides were actually derived from these proteins. This is not as surprising, since the job of a beta cell is to make insulin, but it allowed us to also identify new antigens, particularly relevant, that I will speak about in a moment, such as secretogranin 5, urocortin 3, and proconvertase 2. However, when we looked then to see whether CD8 T cells from type 1 diabetes patients and healthy donors in the circulation were capable of recognizing these antigens, we got again the same answer as before. No difference, irrespective of disease status, and again, a frequency that was in the blood, again, in this quite narrow range. So based on this, we proposed this idea that there is a universal state of benign islet autoimmunity in all individuals. And this is based on the fact that these circulating islet reactive CD8 T cells in type 1 diabetes and healthy donors display similar frequencies, similarly largely naive fractions, and something that I'm not showing you today, but if you isolate these T cells, you differentiate them in vitro into effector memory T cells. Actually, they are similarly capable of killing beta cells, irrespective of whether they come from a type 1 diabetic or a healthy donor. So these observations raise a number of questions, which are now the matter of our follow-up work. And I think the most outstanding question is that basically the bottom message is we are all autoimmune. Then why we are not all diabetic? What is the difference between this benign islet autoimmunity and the progressive autoimmunity that we see in type 1 diabetes? There are different hypotheses we are considering. The first hypothesis, that is a T-cell-centric hypothesis, that perhaps we all have this autoimmune repertoire, but that the regulatory mechanisms that are at play in healthy individuals are more efficient than the ones at play in type 1 diabetic patients. And this fits with the previous observation from ourselves and many others, that when you use a different detection technique to identify these autoreactive T cells, this is a functional assay with an ELISpot, in which you put the T cells in contact with the peptides. If they recognize it, they secrete interferon gamma, you can count the spot. Well, you see that you can readily distinguish the type 1 diabetic patients from the control groups. And this is probably because in this type of assay that is done on all PBMCs, the regulatory network that is at play is maintained in the culture web. So we are now looking more in detail what these mechanisms could be. The second hypothesis, more provocative, and this relates also to this review that we wrote with Desio Eisirik in Diabetologia, is that perhaps the difference between staying healthy and getting type 1 diabetes is in whether the beta cell is more or less vulnerable to this autoimmune repertoire that we all harbor. And there are several lines of evidence suggesting the importance of this mechanism. The first is that when you look for these islet reactive CD8 T cells, the same that we have searched in the blood and that we have found at similar frequencies in type 1 diabetic and healthy donors, you can do this type of staining in situ with tetramers in the pancreas, and we do not see what we do not see in the blood, we see in the pancreas, meaning that these islet reactive CD8 T cells are enriched in the pancreas of type 1 diabetic donors, so suggesting that there are some local factors promoting their homing to the pancreas target organ. The second observation is that the presentation of these antigens via the HLA Class 1 molecules is increased when beta cells are exposed to inflammation. You see here the upregulation of HLA Class 1 when you're exposed to cytokines, which have been known for many years. But this upregulation also results in a higher display of a larger number of peptide antigens, meaning that the beta cell will become more visible to these autoimmune T cells. The third observation is that we observe that among these new antigens that we have identified as exposed by beta cells, some are actually quite recurrent. We have analyzed in the first place the most common variant of the HLA Class 1 molecules, which is called HLA-A2, which is present in about 45% of Caucasian patients and 35% of African persons, a bit lower in Asia. And you see that when you compare with what happens for another variant of HLA, HLA-A3, you see that there are several targets in common. And also that these targets are shared even in the mouse that obviously use murine HLA Class 1 molecules, MHC Class 1 molecules to do this presentation. So suggesting that these antigens are quite important in the pathogenesis. And indeed, this is experiments that we have done when you enrich the presence of these CD8 T cells by in vivo immunization in NOD mice, and then you take these CD8 T cells and you transfer them into empty immunodeficient mice, not SCID mice, you see that they transfer disease very efficiently, similar to what is observed for a reference insulin peptide. And this diabetes is accompanied by destruction of beta cells in red, not anymore visible, and an infiltration of CD8 T cells. So the insulin granule is a rich source of epitopes, but we also observe that not only those are all insulin proteins, but they also share several other features with insulin. They are soluble products that are produced as precursors. So there is a leader sequence that is cleaved in the endoplasmic reticulum, and also they undergo some intermediate processing, like for insulin to release the C-peptide, to release the bioactive molecules. And there is a very well described impairment of proinsulin processing in the islets of type 1 diabetic patients. And so our hypothesis is that this impairment may also impact these other antigens. So this impaired processing may actually divert the digestion, the processing of these antigens towards the pathway of HLA class 1 presentation, and thus increase the visibility of the beta cells. So if we resume all these with a cartoon, there are islet reactive CD8 T cells circulating in all individuals, and this is due to a leaky process of thymic deletion that we have also characterized along with others. However, it is only in the type 1 diabetic condition that these CD8 T cells get activated and are able to migrate to the pancreas when we found them enriched indeed in the patients. So in this scenario, this switch from benign autoimmunity to type 1 diabetes may be critically dependent on two mechanisms, not mutually exclusive and probably of different weight according to age of onset. The first are these T cell mechanisms, the loss of immune regulation that may be more important in younger patients. And the second is this mechanism related to beta cell and with its visibility to these autoreactive T cells. And this may be more important in older onset patients. And this is something that we have presented more in detail in these two back-to-back reviews published recently in Diabetology. And understanding this switch from benign autoimmunity to type 1 diabetes may be key for developing biomarkers of disease progression. You see here the classical natural history of type 1 diabetes, so there may be a gradient from more or less benign to more or less progressive islet autoimmunity throughout this natural history, but also at the clinical stage, this more or less progressive autoimmunity may give reason of different phenotypes related to age, but also these some slow type 1 diabetes cases that we frequently observed in the clinic. This is the people in the lab that has done the work, and our multiple collaborators on this work and our sponsors. And
this is fascinating. Thank you so much, Roberto, for sharing this. You have a large network of individuals all contributing to this work, so that's pretty amazing. I wanted to just talk a little bit about, so what are you doing, I guess, in terms of furthering the cataloging of the HLA epitopes?
Yes, what we are doing right now is two things. The first is that we are looking at techniques to isolate these peptide HLA complexes from beta cells that have more in-depth characterization capabilities. And this is work that we are doing in collaboration with Nicolas Ternet in Oxford, who is an expert in HLA peptidomics. And the second thing that we didn't talk about today is all the issue about neoantigens. What are these neoantigens? are antigens that derive from proteins in the form of peptides like the others, but they're not templated in the genome. Why is that? Because they can either derive from a post-translational modification of proteins, the addition of chemical groups, for example, the citrullination that is very well described in rheumatoid arthritis, or also with this other mechanism, there may be some peptide fragments that fuse together in unexpected ways to give rise to some chimeric sequences that are very difficult to identify, but may be more prone to break the immune tolerance because they are not templated in the genome, so the immune system regards them as foreign rather than self.
And so I guess there's been a lot of talk also about the fact that, mice are so different than humans, but I mean, from this work here, you did show a lot of the overlap with your Venn diagram there. I mean, how would you, sort of further explore that?
Yes, now the things we are doing in that direction is to characterize these T cells that recognize these granule antigens in the NOD mouse. Why is that important? Because we have better access to the pancreas and other organs to see how this works, but also because this can give us other evidence about their pathogenic relevance. For example, we are trying to identify the antigen receptor by which these T cells recognize these antigens. And the idea is to then make transgenic mice in which all of a part of these T cells recognize these antigens to see if these mice develop accelerated diabetes compared to others, which would give another evidence for the pathogenic role of these T cells.
And I guess I'm going to ask one question, then I'm going to open it up to the audience, but I wondered, what are your thoughts on modification of the TCR, in those that, I guess, that have one maybe autoantigen, and then what are your thoughts about potentially modifying the TCR in those individuals? Is it possible? I mean, just sort of speculative.
I think, is it possible in the sense that it will likely be impossible to modify all the TCR repertoire to divert the whole autoimmune response, but this can be used to increase the organ specificity and the homing of T regulatory cells, like many investigators are doing. They're trying to produce Tregs, CAR Tregs, that are reprogrammed to recognize islet antigens, and this should increase their homing to the pancreas and their activation once they get there. So I think that's a quite promising avenue to explore, and that's also where these epitope discovery efforts can be important, because they increase the options that we have for this type of approaches.
Great. We have a question from the audience, Nara Leti. What are your thoughts on HLA class 2 expression in T1D beta cells? Are you planning to perform peptidomics of class 2 as well?
Yes, this is an interesting question, because there has been reports about this expression, and indeed this expression is there, can be detected. Whether this is capable of activating T cells, so would be CD4 T cells, is more questionable, because on one side, I mean, questionable. On one side, they would need co-stimulation to get this activation, but on the other side, if they get to the islets, you may expect that most of these cells are not naive anymore. They've already been activated, probably in pancreatic lymph nodes. They have already some effector memory phenotype. and so maybe less dependent on co-stimulation. So this is something that it will be interesting to explore. The other point is what is the amount of these HLA of the antigen presentation that can be achieved by the low level of class 2 expression that is on the beta cells. So,
and just to sort of extend that, with this is like a deputization, right, of the beta cells to act to express the HLA class 2, and it's transient. It's not just always going on. I mean, is it virally induced or?
Yeah, it's not always going on. Is very likely induced by inflammation or viral infection, and the inflammation that ensues. This is true also for the class 1 to a large extent. There is not much HLA class 1 expression in beta cells in the basal state, but it gets much higher in inflammatory conditions, and this is the case for a number of other cells in different tissues.
So you really need to catch a snapshot of what's happening, right? It's sort of this point of inflammation.
Yeah, this is the other difficulty of why tracing a natural history of disease with histopathological studies like the fantastic progress that EMPOD and other networks have allowed us to do. It is still difficult to do this because maybe it's not a continuous phenomenon, but it's a flare and relapse type of process where you need to be lucky to get the snapshot at the right moment, and it may not tell you all the story. So I think.
And you know, that's reminiscent of many of these other sort of cousin diseases, right? The RA and MS, et cetera. I wonder if anyone else has another question from the audience. You can unmute yourself or you can add something to the chat.
Sure, I have a question. Roberto, thank you for that overview. That was really helpful. My question is, if you compare some of these HLA class 1 epitopes that you presented, and something like Recurrent 3, for example, is about 1,000-fold less abundant than insulin. How does that factor into your thinking as to which ones of these are more important, and particularly early on in the disease when autoimmunity fails to remain suppressed or gets triggered?
Yeah, I think the abundance of the peptide is not always proportional to its importance. Urocortin is a good example, and by the way, as you know, it's also in alpha cells. The other example on the mouse side is IGRP. There has been a lot of work suggesting that this IGRP reactivity is quite important in mice. Yet when you do these MHC peptidomic studies in islets from NOD mice, you barely see any IGRP peptide. So the abundance is a factor that favors the activation of the T cells and the vulnerability, but probably there are also other factors. What are the natural biases of the T cell repertoire in a given individual against one antigen or the other? And the other point is there a cross-reactivity maybe of some T cells that are poised to do something else? For example, there are viral reactive T cells, and they happen to also recognize some self-antigens in the beta cells. So the abundance is not the whole story. The other point is the processing. Urocortin certainly is much less abundant, but it has this very peculiar processing similar to insulin, which may actually favor its export through the HLA under some stress conditions.
Here's a great question from Matthias van Hereth. What about innervation as a key factor in this unmasking process of the beta cells to the immune system? What do you think?
Yes, very good question. The innervation is something that not much known, and I'm not very familiar with this literature. I have to say myself, but it is possible also that what we can call the paracrine factors in the end that are released through innervation can also change the HLA expression. This we don't know, to my knowledge, and also may change the metabolic programming of the beta cells to modify they may also modify indirectly the peptide display that these beta cells give to the immune system.
Yeah, it's very interesting. We recently spoke to Zach Freiberg. He's an MD PhD at University of Pittsburgh, and he was discussing sort of the dopaminergic input on beta cells and alpha cells and the resulting dysfunction that can occur in that context, right? And so then if beta cells become turned off for some kind of insult with the dopamine, alpha cells are now continuing to secrete their product, right? And so then you're getting a high glucose that's toxic to beta cells. Now you're starting to really get into a problem in the landscape there, and then maybe the immune system's called in to sort of mop up some of the problem. So it's a complex question when you start talking about the innervation, but I think it's something that does need to be discussed more often. And I know that the Von Hereth group, as well as Philippe Blanclot's group down in France, also have explored this. So is there any other question? If not, I'm just going to say thank you very much, Roberto. We hope to hear from you again, and we're looking forward to speaking with the Decio Zurich next week. And just sort of, appreciate all the work you've done, the fine work of, really digging into the characterization of these epitopes. It's something that probably needs to have been done for a while now, and it's great that you're actually attempting it and doing it properly. So thank you very much for all your excellent work. And it was great speaking with you.
Thanks for the encouragement. Thanks for the invitation and good discussion. Yeah.
It's great. Bye. Have a great rest of your day.